A very good morning or afternoon to everyone watching. I, Vidhi Rakshe, will be talking about a developing concern, antibiotic resistance. Antibiotics are widely used to treat bacterial infections in humans and animals. However, their widespread and incorrect use has raised concerns about the development of antibiotic resistance. Let us investigate these topics to better understand the potential consequences of antibiotic use. Our first question is, what are antibiotics? Antibiotics are powerful medications that treat certain infections and can save lives when used properly. Antibiotics are powerful drugs that help our bodies ward off diseases caused by bacteria. When used appropriately, they quickly and effectively eliminate infections, causing us to feel better in just a matter of days. However, when used to treat other health conditions, antibiotics are not only ineffective but also harmful to our overall health. Bacteria are microscopic germs that live inside your body, on your skin, and all around you. Most types of bacteria won't hurt you. Some types, like some that are in your gut or in your skin, help keep you healthy. But certain bacteria can make you sick, with the effects ranging from just a mild infection to a severe one that lands you in a hospital. That is why antibiotics are so important. They can help you feel better and are often life saving. Since their discovery, antibiotics have revolutionized modern medicine, saving countless lives and enabling the treatment of once deadly infections. However, due to the overuse and misuse of antibiotics, it has led to the emergence of antibiotic resistant bacteria, posing a significant global health challenge. Using antibiotics when they are not needed, like for viral infections or mild bacterial infections, has created unnecessary side effects and contributes to this global problem. Talking about the history and discovery, the first antibiotic was penicillin. The discovery of penicillin stands as a milestone in medical history. In 1928, a Scottish bacteriologist, Sir Alexander Fleming, observed that a mold called penicillium had killed the surrounding bacteria in a petri dish. Recognizing the mold's antibacterial properties, he named the substance penicillin. However, progress was slow due to limited resources and skepticism. A breakthrough then came in 1938 when a team led by Howard Florey and Ernst Boris at Oxford University successfully isolated and purified penicillin, making it more suitable for mass production. Amid the World War II, American pharmaceutical companies ramped up production, addressing infections in soldiers and sparking recognition of penicillin's civilian healthcare potential. This discovery marked the beginning of the antibiotic era, leading to the development of various antibiotics targeting different bacterial infections. Penicillin's accidental discovery, combined with subsequent collaborative effort, has made a global impact. What do antibiotics treat? Two major types. Two major types of germs can make people sick: bacteria and viruses. They can cause diseases with similar symptoms, but they multiply and spread illness differently. Bacteria are living organisms existing as single cells. They are everywhere and most don't cause any harm and are beneficial. But some bacteria are harmful and cause illnesses by invading the body, multiplying, and interfering with the normal body process. Antibio- Antibiotics work against bacteria because they kill these living organisms by stopping their growth and reproduction. Viruses, on the other hand, are not alive. They grow and reproduce only after they've invaded other living cells. The body's immune system can fight off some viruses before they cause illness, but others, like colds, must simply run their course. So, antibiotics do not work against viruses. So, how do they work? Antibiotics work by targeting specific components or processes within bacterial cells, and they disrupt their growth and survival. Different types of antibiotics have various mechanisms of action. But here are some common ways in which antibiotics function. The first one is cell wall inhibition. Many antibiotics, such as penicillins, interfere with the formation of the bacterial cell wall. Bacterial cells have a rigid cell wall that provides structural support and protection. These antibiotics inhibit enzymes responsible for building and maintaining the cell wall. This causes the wall to weaken and eventually leading to cell lysis or bursting of the cell. The second method is protein synthesis disruption. Antibiotics like macrolides, tetracyclines interfere with the bacterial protein synthesis. They target the ribosomes, the cellular machinery responsible for assembling the proteins. By disrupting the assemblage, these antibiotics prevent the bacteria from producing essential proteins needed for their survival and replication. DNA RNA interference. Quinolones and fluoroquinolones are antibiotics that interfere with bacterial DNA replication and transcription. They inhibit enzymes called DNA gyrase, which are crucial for maintaining the integrity of bacterial DNA. By disrupting these processes, these antibiotics prevent bacterial cells from replicating and dividing properly. Metabolic pathway inhibition. Some antibiotics disrupt the bacterial metabolic pathways by interfering with the synthesis of essential molecules such as folic acid. Bacteria need these molecules for various cellular processes, and inhibiting their production hampers bacterial growth and survival. Cell membrane disruptions. Polymyxins are antibiotics that disrupt bacterial cell membranes. They interact with the lipid components of the membrane, causing it to be permeable or leaky. This disruption of the cell membrane integrity leads to the death of bacterial cells. It's important to note that antibiotics specifically target bacteria and do not have the same effect on all human cells. This selectivity is due to the differences in the structures and processes between bacterial and human cells. What is antibiotic resistance? Antibiotic resistance is a phenomenon where bacteria evolve and develop the ability to survive exposure to antibiotics that were originally effective in killing or inhibiting their growth. This resistance occurs when bacteria undergo genetic changes that allow them to withstand the effect of antibiotics, rendering these medications less or completely ineffective. The misuse and overuse of antibiotics are key drivers of antibiotics resistance and have led to the emergence of antibiotic resistant microorganisms. This is posing a significant threat to the public health and the emergence and spread of antibiotic resistance. Antibiotic resistance bacteria has become a significant global health issue. Understanding the impact on various microorganisms of the antibiotics is crucial to develop strategies for the responsible use and to combat the rise of antibiotic resistance. Some antibiotics that are used to be typical treatments for bacterial infections now don't work as well, and some drugs won't work at all against some bacteria. When an antibiotic no longer works against some strains of bacteria, those bacteria are said to be antibiotic resistant. There are several ways in which bacteria can develop antibiotic resistance. One is by genetic mutation. Bacteria reproduce rapidly and occasionally random genetic mutations occur. Some of these mutations might provide a survival advantage when exposed to antibiotics, allowing the mutated bacteria to thrive while susceptible bacteria is killed off. These resistant bacteria can then multiply and spread. Horizontal gene transfer. Bacteria can share genetic material with each other through processes like conjugation, transformation, and transduction. This means that a bacterium resistant to a certain antibiotic can transfer its resistance genes to another bacterium, even of a different species, making that bacterium resistant as well. Overuse and misuse. When antibiotics are used inappropriately, such as not completing a full course of treatment or using antibiotics for viral infections as they are ineffective against it, bacteria are exposed to suboptimal levels of antibiotics. This can give rise to partially resistant bacterial populations that survive and multiply, leading to the development of a full-blown resistance. Use in agriculture. Antibiotics are often used in agriculture to promote growth and prevent disease in livestock. This can lead to the development of antibiotic-resistant bacteria in animals, which can spread to humans through the food chain or other means. The lack of new antibiotics. The development of new antibiotics has limited over the years and has slowed down in recent years, which limits our ability to find the emerging resistant strains. Antibiotic resistance poses a significant threat to public health as it can lead to infections that are difficult or impossible to treat. It has increased the risk of prolonged illness, hospitalization, or even death. To address this issue, it is crucial to use antibiotics judiciously, finish our prescribed courses, and only use antibiotics when they are truly necessary. Researchers and medical professionals are also working to develop new antibiotics, alternative treatments, and strategies to combat and manage antibiotic resistance. So let's take a step to fight antibiotic resistance. Thank you, and I hope you learned something new.